Hello dear students, welcome. This is Dr. Hadi here and you are watching Medical Globe. Dear, today's topic is from biochemistry, that is secondary structure of protein. We have already discussed primary structure of protein in detail. Now here we go with the secondary structure of protein. Let's start. The definition of the secondary structure of protein is that when you have a polypeptide, I'm expecting you people that you know very well the polypeptide. That is when many amino acids are linked together, they make a polypeptide. So let's suppose each circle represent one amino acid. This is just for simplicity. So many amino acid link, they make a polypeptide. So I'm going to write here polypeptide bond. Polypeptide, not so only is a polypeptide. So for more simplicity, if I write just one line, one single line like this, so no circles are there, still we will assume that this is a polypeptide. Fine. When that polypeptide makes some sort of bend, some sort of twist, like maybe they become like this, it's okay. So, or they become like this, whatsoever, it is also kind of bend, or twist is also kind of bent structure. So whatever structure they acquire, whatever shape they acquire after bending, we call that bend structure as the secondary structure, Now if you take a thread, and you make it like this, a zigzag manner, then there is a chance that it becomes straightened again. So if the protein structure acquire this shape, there is a chance that it become destabilized. We need to stabilize it. We need to maintain this shape in that form. So what we need another bond. We need some special bond that is called as hydrogen bond. Hydrogen bonds are weak relatively, but when many hydrogen bonds are collected together, then it will give us a strong structure. So by the help of hydrogen bonds, that structure will remain maintained. How? Let's suppose if I assume that there are amino acids in that structure, or here whatsoever where you can see there are amino acids. In between the amino acids, there will be a hydrogen bond. So here is hydrogen bond. Here is hydrogen bond. One amino acid linked with another amino acid by the help of peptide bond. Okay, we already discussed it. We know very well. But there is another bond, second bond comes, that is hydrogen bond between one amino acid and another amino acid. And we say a stabilized structure has been formed. Okay, fine. Now, so I think we should write when a polypeptide bends or twists. So here we should write it is also stabilized by the help of hydrogen bonds. Now it's fine. Now the definition is okay. When the question comes in exam, what should you write first? The definition within 2 lines, that's enough, gone. Then immediately if it is followed by the types of the secondary structures. There are 2 types of secondary structure. One is called the alpha helix, and the second one is called as beta pleated sheet. So start with alpha helix. When a polypeptide bends itself and it gets the shape of a helical form, means like this is called helix, something zooming like this, right? So that helical structure, helix, the most important thing that we should know, that we should write on the paper, is we have some points that we should know. The first point is that there are turns in the alpha helix. And let's suppose if I start here, from here and end here. So this become one turn and then second turn and then like this. Okay, this is one turn, second turn, third, fourth, fifth, sixth, seventh, and so on. There are many turns. In each turn there are 3.6 amino acids in each turn. This is the first point that we should remember. What is mean by 0.6? If you say 3, it's okay. 0.6, that is a little bit confusing. This means that there are 3 amino acids in that turn. The fourth amino acid is partially present in that turn. Its remaining part is present in the next turn. So it is not, the fourth amino acid is not fully present in one turn. That is why we say 3.6. Mean if you divide one amino acid into 10 parts, then sixth part will be in one turn and the remaining part will be another turn. That's why. Okay, now this is the first point. Second, how the amino acids are linked with one another by the help of hydrogen bond. This is so interesting, and a little bit complex. In order to make the topic easy, I don't feel the need to write all the amino acids in the secondary structure. I will assume that these are amino acids. Okay, dots. But I will show only 2 amino acids. That will be enough for us to get the idea. Okay, so let's suppose we start the turn like this. If you move like this, you will encounter with the first amino. Let's suppose this is the first amino acid. And inside one amino acid, we know very well that there is an alpha carbon, NH group. It has made a bond, so it is not NH2, it is just NH. This is one amino acid. This is C double bond O, and there is another amino acid here. This is another amino acid, alpha carbon, C double bond O, NH, and it is also linked with more and more amino acids. So this is the bond. This is the bond, we call it peptide bond between the C double bond O and NH. If I show this is one amino acid and it is C double bond O linked with carbon, this is the bond linked with which carbon? Alpha carbon. And then this carbon is linked with the NH group. So I'm just going to write here NH group. Now there will be another amino acid here. There will be amino acid here, like this here. You can see there is another amino acid. So I'm not going to write the complete structure. So if there is another amino acid, then this line, that blue line, that now I made it dotted line, this must represent the polypeptide bond. This must represent the polypeptide bond. So if it is amino acid, this must represent the polypeptide bond. Now, if you move on that side, then you will see another C double bond O, and of course an alpha carbon, and that alpha carbon linked with the NH group. So this is next amino acid. So if this is one amino acid, it has made a peptide bond with that amino acid, and it also has made a peptide bond. So I'm going to write it dot. So here is another amino acid. Now you can write like this, but I don't want that the structure become complicated. My interest is with that amino acid only. Suppose we are going in that direction. Suppose if this is the first amino acid, and you go, this is second amino acid, and then you go third amino acid, and suppose there comes third amino acid, and there comes the fourth amino acid here. I'm not going to write the whole structure, you know very well. And here comes the fifth amino acid. Let's suppose this is the fifth amino acid. Let's suppose this is the fifth amino acid, where you can see there is C double bond O, alpha carbon, and the NH. So this is the fifth amino acid, and this is that was the first amino acid. What happened? The hydrogen bond is formed between the C double bond O oxygen of one amino acid with the hydrogen of the NH group of the next amino acid. So here a hydrogen bond will be seen. In this way, all other hydrogen bonds, they will also be mentioned in the same way. So with this one hydrogen bond, my students, I think, got the idea of how the hydrogen bonds are made. But remember, the number is also important. If this is assumed to be the first amino acid, then it will make the hydrogen bond with upcoming fifth amino acid. So 1, 2, okay. If you move like this, move like this. So this will be first, second, third, fourth, and fifth. Okay, fine. If this is second amino acid, let's suppose this dot is second amino acid, so it will make a bond with upcoming sixth amino acid, like this. Let's suppose this is the first amino acid, second, third, fourth, fifth, sixth, seventh, and so on. The first amino acid is always linked with the fifth amino acid. The second amino acid is always linked with the sixth. The third one is with seventh, and so on. So this will be the style. First amino acid linked to the fifth amino acid, and second to the sixth, third to the seventh, and so on. You can go on. So this is so lovely structure of the secondary structure of protein, and how the hydrogen bonds are there. And one thing I would like to tell you, that this structure is stabilized by hydrogen bond. It is in that form. There are some naughty amino acids which, if present in that alpha structure, they will disrupt that structure. And do you know who's the name of that amino acid? What is the name of that amino acid? That is proline. And according to in some books also, glycine is also there. But proline is so prominent. The presence of proline will make some kind of bend in that structure. So this point is important. This is finished now. Come to the next one, that is beta pleated sheet. In beta pleated sheet, once again we have a polypeptide chain. One or more than one polypeptide chain. And they are folded just like you fold cloths. You have your cloths, you have some covering sheet you have, and then you fold it like this. Okay? Just like some students living in the hostel, and they don't living in that home and living in hostel, so every morning, every time they have to fold their goods like they are very expert. So here in beta pleated sheet we have polypeptide chain, and move like this, and then bend, then move, and like this. And there will be, of course, amino acids will be there. So I'm going to write just circles. I'm just going to write the circles. Each amino acid will be linked with another amino acid by the help of hydrogen bonds. Now there is one word, interhydrogen and intrahydrogen bonding. What is the difference between these two? If there is one polypeptide and it has make this kind of folded structure, then the hydrogen bond here within that structure will be called as intra. I will use the word intra chain hydrogen bonding. And if there are more than one polypeptide chain, and let's suppose I'm right, this is one polypeptide chain, this is another polypeptide chain, this is another polypeptide chain, and there are many polypeptide chains, then of course there will be amino acids, and the hydrogen bond will be formed just like there, but this time the hydrogen bond is called as interchain. So intra and interchain amino acids. This was just simple structure of the beta pleated sheet. Now if you want to go into the depth, this is enough Still, if you want to go into the depth and you want to write the structure of the beta plated sheet in the detail, original structure, so come, you are also welcome. In order to write the original structure, beta pleated means that a structure that is telling us, that is showing us the amino acids and style, the way they are arranged and how the hydrogen bonds are made. You need to write like this. Can you do it like this for me? Like this. Can you do like this? Everyone say yes, sir. This is so easy to write as a zigzag line. This is so easy. Fine. And I'm sure you can also write like this, one more line. Okay. Fine. I'm also happy. This is the first zigzag line, this is the second zigzag line. You just need to add one more line to that zigzag at the bottom one. Just add one more line here. Just add one more line here. Now what you need to do, you know very well that in one amino acid there is an alpha carbon and there is C double bond O. You know very well. Okay, fine. This is one amino acid and this is NH. This is one amino acid. So how to manage these amino acids, how to write? If I just write just one circle here, this is, suppose this is alpha carbon. This is alpha carbon. Then leave two corners and add another circle here. Again leave two corners, add another circle here. So you got the alpha carbon of all the amino acids. Now this is alpha carbon. Now you can make it arrange. If this is the alpha carbon, let's suppose this is the alpha carbon. So you are going to make this one as C double bond O. Of course, yes, because on one side there is C double bond O. So this must be C double bond O. And we know very, very well that one amino acid is linked to another amino acid by the help of peptide bond. And whenever there is a peptide bond, there will be an NH. There will be an NH. And it is also linked with the alpha carbon, and then there is C double bond O. So this is another amino acid. Now, taking this sequence, if this is the alpha carbon, this is alpha carbon, this is C double bond O, this is C double bond O. Now what comes after that C double bond O? There comes a bond. So this will be our peptide bond. This will be our peptide bond. And if it is a peptide bond, so what comes then? It is NH. So I will write here NH. It becomes so easy. And what comes after that NH becomes that alpha carbon. So this is already alpha carbon. And then what comes here after that alpha carbon? We have C double bond O. We have C double bond O. And then this is again a peptide bond. This is again a peptide bond. So I'm going to write it like this, black line. It's again a peptide bond. So if it is a peptide bond, it must be NH. And then it is another alpha carbon. It is another alpha carbon. And this will be C double bond O. This is finished. So easy now. You can also do the same arrangement with the next zigzag line at the bottom, that is below. You can start the same, means add one circle here and leave these two corners, then add another circle here, leave these two corners, add another circle here, leave these two, and so on. Now if this is the alpha carbon, so what comes after that alpha carbon? That is, we have C double bond O. It is C double bond O. And then what comes? It is the peptide. It becomes peptide bond. If it is peptide bond, then it becomes NH. It becomes NH. And then it is alpha carbon, then it becomes C double bond O. If it is C double bond O, then it becomes peptide bond. If it is peptide bond, then it must be NH. And if it is NH, then it will be alpha carbon. And if it is alpha carbon, then it will be C double bond O, and so on. Wow, this is so easy. Means we have mentioned every amino acid in the beta plated sheet. All amino acids, their arrangement, the proper arrangement in detail, original structure is there. So now the hydrogen bond is always formed between the hydrogen of the NH to the oxygen of the C double bond O group. So here this hydrogen, this is so easy now. This oxygen partially negative charge, this oxygen has partial negative charge. So it will make hydrogen bond with the partial positive hydrogen. This is one hydrogen bond. And you can also make this hydrogen will make a bond with that oxygen. So in this way we got the beta plated sheet. This is one polypeptide and is folded another polypeptide. There's a hydrogen bond and we've got beta plated sheet. They say where these structures are found, usually both alpha and beta structures are found inside protein, but the beta plated sheet is found in the tertiary structure of protein. Especially they made the core, I mean the middle part of the protein is usually made by this beta plated sheet. I hope you got the idea. If you start writing these on a page, you will fill almost 2 to 3 pages if you got all the idea. I hope you enjoyed watching that video, and also you got that video lecture. Thanks for watching that lengthy lecture. Remember us in your prayers. Take care. Bye bye.